episode is meiosis. So let us start. So at the end of the lesson, we will be knowing about what is meiosis. Now in your 10th standard, you don't have the basic stages, all the stages of meiosis in details. You are having only the basic information of meiosis. I have tried my best to make it in a very simple way. I have shown the pictorial representation of this meiosis. You will be knowing the meiosis and different stages through different pictures, stage by stage pictures. We will know what is a homologous chromosome and how it is formed, and we will also know the significance of meiosis. So let us start with today's topic. Now here we have started with meiosis. Now it is a basic introduction. We will be discussing in details in the following lessons. So all the mitotic cell division takes place in somatic cells. Remember that. Now here we are starting with meiosis. So it takes place in the reproductive cells like the gametes, sperms and ova. Now it is a modified form of mitosis in which the chromosomes divide once while the nucleus will divide twice. Now remember this, the chromosome is dividing once whereas the nucleus is dividing twice. Now the number of chromosomes are reduced to half. Here we have started with 2n number of chromosomes, with which at the end of the meiotic 2-cell division they will be reduced into haploid number of chromosomes. Now 4 cells resulting from a meiotic division have a haploid number of chromosomes, and the chromosome number becomes half in each cell as discussed. So it is starting with 2n number of cells and they end up with the haploid number of cells. So that for that reason only it is called a reductional division. So this is a overview, or we can say that it is a diagram of meiosis which is forming into 2 stages. Now the first stage we will highlight, it is the homologous pairs are separated and they are reducing chromosome number into half. Now in meiosis 2 what is happening? The sister chromatids are separated and again they are producing 4 haploid gametes. So this is what is happening in the meiosis 1 and meiosis 2. Now meiosis 1 again is divided into 4 phases: prophase 1, metaphase 1, anaphase 1 and telophase 1. Now in prophase 1 only the crossing over and all will take place, which we will discuss in details in the coming slides. And here this meiosis 2 is exactly the same as what is happening in mitosis. So if you have not gone through my mitosis video, I'll put a link up here. Please go through that mitosis so that you can get to know what has happened in mitosis and you can relate it with the meiosis 1. So let us move on to the next slide. Now here we will be discussing about homologous chromosome. Now this is quite a new term to you all. I think you don't know what is homologous chromosomes, but we will go through this term when we are starting with the meiosis steps, meiosis 1 step. So first meiotic division is having this term, homologous chromosome. So what is it? Let us have a look in this. Now this is a homologous chromosome. What is here? There is a pair of chromosome from which one chromosome is coming from father and another chromosome comes from mother. So they are not identical. So here we have discussed with 2 different colors. So if green chromosome is coming from father, the purple one is coming from mother. So they are similar in length, their gene position and both the location of the centromere also you can see is the same, but they are not identical. The gene, however, this gene contains different alleles. So this all we will be discussing in detail in the next chapter when we will be studying the chromosome. We will have a lesson where we will study in detail about chromosome, about the alleles and all. We will study them in heredity lesson. But here let us have an idea about what are homologous chromosomes. So this is a homologous chromosome, and how does it look like? Having 2 different chromosomes, one from the father and one from the mother. So they are not identical, and their position, length, gene position, location of the centromere is same. So you can see that after replication, this homologous chromosomes, they look like this. So they are having sister chromatids. So same like chromosome, they have produced one. So this is only replication. So they have produced their own replica. So if this is coming from the father, it has produced another sister chromatid. And if it is coming from the mother, it has produced another sister chromatid. So these are homologous chromosomes. So I think it is clear to all of you. So let us move on to the next one. Now in this slide, I have taken another pictorial representation. Now here we will see meiosis and crossing over. As you don't have the stages in details, I have not written those. But let us have a quick idea of those. Now previous slide I have discussed about the homologous chromosomes. So here see the homologous chromosomes, one received from the father and one received from the mother, I have already told. They come in line pairs. So we have seen the pairs there. Now here what is happening? Now it is meiosis 1, where the chromosomes continue to shorten and thicken. And each chromosome they split lengthwise into two different chromatids. We can see, we have seen there the homologous chromosomes after replication, they have formed with sister chromatids have been formed. Now here we have seen they have shortened themselves. They are not yet bound together through the centromere point. So they split lengthwise and into two chromatids, separate chromatids. Now suppose this is from the father, these are two separate chromatids, and this is from the mother, these are also two separate chromatids. Now this again, what is happening? The non-sister chromatids of a tetrad, they break open and rejoin each other. Now here this four separate chromatids, what are they called? They are termed as tetrad. So this is a new term. Previous slide we have learnt a new term, that is homologous chromosome. So try to remember and recall all the terms. Here we are learning a new term when the sister chromatids, they break open. The individual chromatids are called tetrad. Now what happens in the next step? This non-sister chromatids or tetrad which break open, they rejoin each other. See, you can see one is red and one is white. So they are rejoining, and this rejoining is called crossing over or chiasmata. Crossing over or chiasmata. So exchange of some genes or some portions of the chromatids takes place between the paternal and the maternal. So both the father's genes and the mother's genes are mixing here together. So paternal and maternal gene exchange takes place here. Now the pair of homologous chromosomes, it is happening all during meiosis 1. So this phase where the pair of exchange of maternal and paternal genes are taking place is called crossing over. Now what is happening due to crossing over of homologous chromosomes? The nucleolus and the nuclear membrane, they disappear. They start disappearing in this stage, and the membranes of homologous chromosomes, they completely separate each other and they move towards the opposite poles. The nuclear membrane then again starts reappearing and leading to the formation of two daughter cells. Now here the crossing over takes place. Then they all are arranged in the equatorial region. Now in the similar way, if you can recall the mitosis stages, they will all separate from each other and they'll all move towards the pole. Then again at the last stage, what will happen? The nuclear membrane will start reappearing and they'll form two daughter nuclei. So this is all what is happening in meiosis 1. So what will happen after these two daughter cells are formed? Then comes the meiosis 2, or it is actually similar to mitosis. Same things will happen, and during this stage the two chromatids of each chromosome, they separate, they move towards each poles, nuclear membrane reappears and the four cells are formed. So this is daughter nuclei 1 and this is daughter nuclei 2, and so again the daughter cells are formed, the nuclear membrane reappears, and the 4 cells which are formed are having haploid number of cells, that is n. So its original cell started with 2n, and here it started with 2n number of cells. But after full meiotic division is complete, we can see here we are having only n number of cells in each cell. So I think it is clear with everyone. Is it clear? So I can't see your faces, neither can I ask you questions here. So what I want is your feedback. I want you to comment. I want you to ask more and more questions so that I can understand how much you are understanding. And this meiosis and crossing over is a bit complicated process. What is happening? If you are not clear in the first stage, when we start with the new lessons, it will be quite difficult for you. So please be clear. If you are not getting anything, every time I keep on repeating the same thing, please do ask your query so that I can clear it up in the next topic. And please do subscribe to my channel and don't forget to like and share so that all your friends can also get the chance to learn from my videos. So let us see what is there again in the next slide. Now in this slide we will discuss about crossing over or chiasmata. So I have told in the previous slide also, during meiosis 1, the non-sister chromatids or the tetrad break open. These are the sister chromatids. They'll break open and they'll form a crossing over, or like parts of this chromosome will combine with this one and parts of this chromosome will combine to this one. So what is happening here at the end of this recombination actually is happening between the 2 strands of chromosomes. So this is called the crossing over. So after crossing over, how the chromosomes are looking, see here you can see this part and this part, they have crossed over. So the blue has gone to this strand and the purple has come here. Here also the same thing has happened. The blue of this part has gone here and the purple of this part has come here. So we have marked these portions as capital A, small a, capital A, small a. So we can see that after crossing over, what has happened? Capital A here and again small a here. So this is after recombination of certain genes. So this is called the crossing over or the chiasma. And this process is called the chiasmata formation. So this I have discussed in the previous slide how it is forming. So this is actually a clear pictorial representation I wanted to give so that it will be easier for you to understand and you'll have a clear picture of that in your mind. So for that only I gave you this. So let us move on to another representation of meiosis. So this is a quick revision of what we have studied till now. So bit complex process, divided into 2 parts, as in all slides I have told you that it is divided into 2 parts. One is meiosis 1 and another one is meiosis 2. So what is happening from the beginning? We have told that the chromosome continues to shorten and thicken. Then crossing over has taken place here. Chiasma formation has occurred. Then portions of chromatids, they'll be separated from each other. Their homologous chromosomes will separate out and they'll move towards the pole, and then the nucleus is also dividing, the cell cytoplasm is also dividing into 2 different cells. So 2 new different cells are formed. Again from those 2 new different cells, same process will continue to form 4 different cells having the haploid number of chromosomes. So this is similar to that of mitosis. So all the chromosomes are arranged in equatorial region. From there the centrioles we can see in the opposite poles. The spindle fibers are formed, as if you remember mitosis, I think you remember, and if you have not gone through those slides of mitosis and cell cycle, Please go through it, and I am giving the link up on this slide so that you can study the mitosis lesson. Now again, these cells, the nuclear membrane, they starts reappearing, and the cytoplasm also divides into two to form 4 different daughter cells having haploid number of chromosome. So this is all about meiosis. Now we should learn what is the significance or importance of meiosis. Let us move on to that. Now this last slide we will learn about the significance of meiosis. So what is the significance, importance? So we have seen the number of chromosomes is reduced to half in daughter cells. First significance. What happens? The result, which results in the formation of haploid sex cells, and which after fertilization they restore the diploid number of chromosomes in the zygote. So this is a main importance, that after fertilization they restore the diploid number of chromosomes in the zygote. Now again, next, what happens during crossing over, which occurs generally in meiosis? What is happening? The parts of the chromatids we have seen are being exchanged. I have shown you in the previous slides with 2 different colors. One is blue, one is purple. So parts of the chromatids, if you remember, are exchanged. So which bring about variations in the offsprings. So if there are 4 siblings, they will have variations. So variations are there in the offsprings. Next importance is 4 chromatids of homologous pair of a chromosome are passed on to 4 different daughter cells. This also causes gametic variation. So all will look different. All the siblings will have the skin color different. So there will be gametic variation. And it also prevents the multiplication of chromosomes, and hence maintains the stability of the species. So this is all about the main significance of meiosis. So as of now, this meiosis or the reductional division is complete. You have seen that it is having many importances also, or significances also. So please go through it. Please go through your textbook which you are following. I think you will get to know everything. And if you are having still any doubts, please do ask me in my comment box. And don't forget to subscribe. I am telling it again so that the next update you will get it as soon as I post it. So for today, the meiosis reductional division is completed. Thank you.